there were radios, and they translated in 7 different languages. And so we were the hotel was right next to the beach, and when you went out to, you could go out on the beach in your swimsuit and get a nice tan and stick a plug in your ear and hear the lecture. And that was all good fun until it came my turn to give a talk. And the auditorium was absolutely empty. And it was the most disconcerting thing, because normally what happens when you're giving a lecture, you watch to see how many people are going to sleep, how many people are taking notes. There's some sort of feedback and so on and so forth going on. So anyway, I'm glad some people are here. In the last row, if you can't hear me, would you go like this? If my voice drops down, or signal me in some way that so I know that I'm losing you or you're losing me, whichever way we want to put it. The title of this talk is, Can the Neurosciences Inform Evolutionary Biology? And I'll start off a bit by reading some points and then get to a freer discussion. Historically, evolutionary biology is focused on behavior, its function and adaptive value, speciation, biodiversity, natural selection, proximate and ultimate mechanisms, and relationships between genes and traits. Inferences have been made about how the behavior works to produce behavior, or how the brain works to produce behavior, but seldom have brain-behavior relationships been a central concern until the development of evolutionary psychology. Now, historically, the neurosciences have focused on the anatomy of the nervous system, the function of different brain areas, brain chemistry, and more recently, the brain's genetic makeup. Over the last 30 years, some neuroscientists have shown interest in relating their findings to those behaviors that concern evolutionary biologists and asked questions similar to those that evolutionary biologists have tried to answer. The amount of research currently being conducted in the neurosciences exceeds that conducted by evolutionary biologists probably by a factor of 10. This ratio is unlikely to change in the near future, and thus it's worth asking, can the findings from neuroscience inform our evolutionary explanations? And the first overhead right here shows some of the possibilities. If we assume that the makeup and functions of the brain are subject to natural selection in the same way as morphological traits, which was Darwin's argument, of course, one might expect that the first possibility on this list is most likely. Many neuroscience findings will be consistent with those from evolutionary biology. But you can see here, or at least I can see a little bit, basically, well, let me get to where I have a copy of this overhead. Well, I don't. These are 4 possibilities. They can be consistent with, like evolutionary biology postulates or posits brain differences in the sexes, and neurosciences could find that. They could be inconsistent with it. They find no differences in the brain. They could raise new kinds of questions. I'll be talking about memory in just a moment. It turns out that memory is terribly inaccurate. If that's the case, and that's the case consistently with most of the studies, then the obvious question is, has this kind of thing been selected, or is this just a kind of quirk of evolution? And then there's the possibility that all kinds of findings from the neurosciences might not be relevant. For example, the chemical makeup of the cerebral spinal fluid, which is essentially the brain's garbage can, one might look at it that way, might not be of any great interest to evolutionary theorists and thinking in evolutionary biology. Anyway, as I said, the title of this talk is, Can the Neurosciences Inform Evolutionary Theory? My answer to the question is yes, but the process will be slow, difficult, and time-consuming. And there are many reasons for that. Could I have the next overhead, please? One is the orientation of many neuroscientists. most of us don't ask questions that are related to evolutionary biology. Thus, the findings are very difficult to interpret in an evolutionary sense. Cerebral spinal fluid, or CSF, is again a perfect example. There have been endless studies, maybe 20,000 studies on the composition of this particular fluid, which is the fluid which surrounds your brain and surrounds your spine and sort of lubricates it. And but only maybe a half a dozen of those papers relate. They've tried to ask whether or not there's an evolutionary importance or significance to it. Second is methodology. Neuroscientists use generally PET, MRI, molecular genetics, gene manipulation, electrophysiological monitoring, transcranial stimulation, electrode implantation, I can go on and on and on, chemical analysis, and so on and so forth. And while these techniques are refined continually, there are limits to the kinds of questions they can answer. Third point that Professor McDonald talked about this morning, although he wasn't emphasizing it, is there's no consensus among evolutionary biologists about what is evolutionary theory. There's just a lot of overlapping. I mean, they're in the same ballpark, as we would say. But there's not necessarily agreement. For example, whether or not group selection occurs, one of the points that was brought up this morning, is still a debatable subject. And then finally, some evolutionary biologists don't read in the neuroscience literature. And that is unavoidable and perhaps unfortunate. Next slide, please. So let me start out with memory. One of the key points about memory, well, the way this slide works is you see time going down. Here's an event up here, such as you focus on me talking for a moment. There's a short-term memory, which I'll get back to in a little while, and brain cells type A and brain chemicals of type A prime do the recording there and so on and so forth. Now, a half an hour later, you're trying to remember this particular moment, and all of a sudden what you find is that there are different brain cells that are reconstituting the memory and different chemicals that are involved. It's not the same system. Now, these differences might seem unimportant. For example, by analogy, you can use a copying machine to duplicate a photograph, and you may get a near exact replication of the photograph, even though the scanning, chemical, and printing processes are entirely different than those that produced the photograph originally. But what is important is whether memories are accurately recorded and whether or not they're accurately reconstituted. This happens rarely. And I'll return to this short-term memory, which was discussed this morning also by Professor McDonald in a while. What I want to focus on at the present are reconstituted memories. That is, let's say a week from now you're trying to remember what these lectures were about, or what you ate for lunch today when you were here, this kind of thing, and that. What happened yesterday, several hours ago, whatever. Next overhead. What this overhead shows is, or are, the sources of memory alterations or reconstitution alterations and so on and so forth. Time, again, flows in this direction. There's the initial memory. Let's say it's 6 o'clock now and you're trying to remember what happened today in these lectures. Your stress level, your mood, your prior experience, your familiarity with your lack of sleep, well, that wouldn't apply until tomorrow morning, but all of those factors will, in fact, influence memory. Further, if you remember something at 6 p.m. today, say again this lecture, or these lectures today, and then you try and remember it tomorrow, there will be further reconstitution, and this just continues. I've listed only stress there, but all the other factors can come in to play. In literally every study that goes on, memory turns out to be not totally wrong, but inaccurate and subject to all kinds of changes as time goes on. So what do we have here? We have evidence from the neurosciences that mood, stress level and duration, prior experience, social context, lack of sleep, familiarity with what is experienced, as well as the number of times you remember something, each of these can have memory-altering effects. In addition, there are other factors that affect memory, such as dementia, neurotoxins, such as alcohol. Most of us have had too much to drink one time or another in our lives, and it's a little hard to remember the conversation towards the end of the evening. Normal aging affects memory. Genetic factors, such as those that contribute to attention deficit disorder, where memory capacities differ significantly from normal. Altered memories, changed memories, inaccurate memories are particularly likely excuse me, when recalling once-only events, such as a rapidly occurring events such as an automobile accident or a bolt of lightning hitting a tree and so on. And let me suggest you think about testing this for yourself. Go home, sit down, see something on the radio, or the TV or something, just record it, hide it. A week later, remember it. A week later, remember it. A week later, remember it. Then compare what you remember, and you'll be astonished at two things. One, what is faded in terms of the detail of the first memory, and two, lots of little inserts that have been added after the event. It's really quite striking, where it only takes about 2 minutes to run that experiment with yourself. With familiar items, such as remembering the names of one's friends, one's dog, or one's telephone number, we seem to remember more accurately than these kinds of once-only events. However, these kinds of memories are memorized. Again, a point mentioned this morning, and they involve yet another brain system, other brain cells, other brain chemicals, and in other functional areas of the brain. Moreover, familiar items are often associated with memory cues, which increase the likelihood of solving inaccurate memories. For example, I drive home tonight, I go in the house, and I set the keys to the car, I think, on my desk. In the morning I get up and they're not there. Well, I know I didn't go back to work because the car's there. My wife can tell me I didn't go back to work, so on and so forth. And so there are only a certain number of places that my car keys can be, and I'll find them in a while, or I usually can find them in a while, and so on and so forth. With a situation where, let's say, you see a bolt of lightning hit a tree, or you see an automobile accident on the way home, these memory cues aren't as available. You can't really retrace in the same sense. Still, even the names of close friends are sometimes forgotten, and at times most of us can't remember where we put our glasses. That's me. Appointments I've scheduled, that's me. And where I left a cup of coffee a few minutes ago, that's also me. Hopefully it's not only me in this group. So let's ask, what have the neurosciences demonstrated about memory? First, memories are vary in their accuracy. Second, they are altered in the process of recall. Third, numerous factors, and I've mentioned those, and they're listed here, interfere with accurate memory. Fourth, we are seldom aware of our memory alterations unless we experience trouble with our memory. And I've set the keys on the desk as I remember it when I come home. So I get up in the morning, I dress, I start to go off to work, and I go to pick up the keys. And only then do I realize, because they're not there, that my memory is a bit fuzzy. Again, sample yourself from time to time. I provided you with some inaccurate information, which I wish to correct. They informed me that even though I earlier said I wasn't an extrovert, I am, and so that issue is now corrected. Okay, now we turn to memory models and strategies. This talk began with a discussion about altered memory, and the question was asked, does altered memory make evolutionary sense? By way of offering an answer to that question, let me first review some findings from short-term memory. Again, a topic that was talked about, and so there's just a bit of review right here. What this slide basically shows is this, that, or here's the way a research or an experiment would go. You show 12 objects on a screen, unrelated objects, let's say, not the various parts of an automobile, but unrelated objects. You put them on the screen, and they're shown there briefly. You have time to look at them, let's say a second, not a long time, and so on and so forth. And what happens is then you ask people to recall immediately what was there. And they'll say a basket and a handkerchief and a funny-looking face and a pencil or something like that. 1 in 100 people can recall 8 objects. The average is from 1.5 to 5 among human beings, and this is consistent and remains consistent across all kinds of different protocols. Whether you're using visual stimuli or auditory stimuli, let's say you give a series of letters or numbers or something like that, or even words, and you pop 12 of them out or 8 of them out and so on and so forth. People are consistent. So it appears that that part of the brain, which is holding the immediate memory, again, you see a car crash out of the corner of your eye, or a lightning bolt hits a tree, and so on and so forth, that just a small part of that total event is getting recorded. So for most of us, most people, it's easy enough to remember 2 or 3 objects if 12 are on the screen. As I said, only 1 person in 100 can remember as many as 8. The majority of individuals fall in the range of 1.5 to 5. And as I said, the number remains highly consistent across different research protocols. Interestingly enough, mood, stress, lack of sleep don't seem to affect short-term memory in the same sense that they affect reconstituted memory. Remember, we started this talk with reconstitution, and there were all these factors that were affecting it. It doesn't seem to affect short-term memory. It probably does, but it would be hard to demonstrate and find. Now, short-term memory is the first step in storing and transferring visual, auditory, tactile information into long-term memory storage. And again, let me suggest an experiment you can do with yourself very much. You can close your eyes